Okay, welcome everybody to our discussion on the respiratory system. This is chapter 13 out of your textbook, and what we're going to talk about are the organs of the respiratory system, the purpose of the respiratory system, conditions that affect the respiratory system, and we'll talk about how the respiratory system works. So, what you're looking at in this figure is just a diagram of the respiratory system, and it consists of the nose, the pharynx, the larynx, the trachea, the bronchi, and the lungs. Now, the functions of the respiratory system are gas exchange between the blood and the external environment, which occurs in the alveoli of the lungs, as well as passageways into the lungs that purify, humidify, filter, and warm the incoming air. And what you're looking at here is a cross section through the first part of the respiratory system. This is the nose. It's the only externally visible part of the respiratory system. Air enters the nose through the external nares, which are shown right here. And once it gets inside, what you'll see are that there are ridges and grooves inside the nasal cavity. The ridges are the superior, middle, and inferior nasal conchae, and the indentations, the grooves, are the nasal meatuses, which are the superior, middle, and inferior nasal meatus. The nose is also covered with a mucous membrane that obviously produces mucus and is also covered by guard hairs called vibrissae. And the purpose of the mucous membrane is to humidify the air, but also to help filter the air that's coming into the nose. Particulates get trapped in the mucus and also trapped by the vibrissae, and the air gets humidified as well as warmed up, and this prepares it for entry into the pharynx, which has three regions: the nasopharynx, the oropharynx, and the laryngopharynx, and from there for entry into the trachea through a hole that's called the glottis. Okay. Now, other features that you'll notice in this drawing of the sagittal section of the head include the external and internal nares. That's the entry and the exit to the nose. This is the nasopharynx, the first part of the pharynx, which is the common chamber shared by air, food, and water. And this opening here is the opening of the eustachian tube, which actually connects the middle ear to the throat. And the purpose of this tube is to regulate pressure between the inside and the outside of the eardrum so that it can vibrate when it encounters noise, which is essentially a pressure wave propagated through the air. Occasionally what can happen is that bacteria can move up this tube into the middle ear and produce an infection in the middle ear known as otitis media. Those bacteria, as they grow, can produce gas, causing the eardrum to bulge out, and this produces tremendous pain. And in the old days, the fix for this was a course of antibiotics and also to lance the eardrum in order to relieve the pressure. And then if this was a recurrent problem, we would use myristogomy tubes, basically plastic tubes with sleeves on them that we would insert into the eardrums that would regulate pressure on either side of the eardrum. And what that did was made the pressure equalize. It also brought air behind the eardrum, and the bacteria that like to grow in the middle ear that produce otitis media are a little bit resistant to oxygen. They like a more anaerobic environment, so this can help not only relieve the pressure but relieve the possibility of the bacteria growing. These days they don't go to lancing the drum. They try and treat primarily with antibiotics in an effort to kill the infection. The air, once it swirls around inside the nasal cavity, exits the nose via the internal nares. It'll enter the nasopharynx, will pass into the oropharynx, shown here, and then, because this flap called the epiglottis is in the open position, it'll pass through this opening called the glottis, past the vocal cords, which you can see right here, and then down into the trachea. And so far, all we've done to the air is filter it, humidify it, and guide it down to the lungs. the important part of the respiratory system, which is the respiratory division of the respiratory system that actually begins with a structure called the respiratory bronchioles, is where the gas exchange is going to take place. That's where we swap oxygen and carbon dioxide. We get rid of the carbon dioxide when we exhale, and we load oxygen into the blood when we inhale. And the purpose of that is to provide oxygen to all the cells in the body via the blood that leaves the lungs, heads back to the heart, and then is pumped by the heart to the rest of the body. Okay, so the conducting division that we're actually describing right now consists of the nasal cavity, the nasal pharynx, the oropharynx, the larynx, the trachea, the bronchi, and their branches, which are basically a series of pipes that branch off of the main pipes that go into each lung, called the primary, secondary, and tertiary bronchi, all the way to what we call the terminal bronchioles. And that's just essentially a very fancy pipe. It's called the tracheobronchial tree, that's designed to do nothing more than to get the air to every square inch of the lungs. It's where the gas exchange takes place that we really get the value out of the respiratory system. And while we're on the topic of the respiratory system, we should address why the respiratory system developed in evolution. And the reason is that as life on planet Earth became more complex, moving from single cells to multicellular forms of life to tissues, the organisms became so complex and the tissues started to carry out so many different roles that those tissues weren't necessarily near where the oxygen would come into the organism. And so we had to develop a method to get the oxygen to those tissues, and that organ system that did that was what we call the cardiopulmonary system, which consists of the upper respiratory, the lower respiratory, the lungs, the heart, the blood vessels, and the blood that's contained in it. And this allows every cell an opportunity to access the life-giving oxygen that it needs in order to make enough ATP to run the body. Okay, so that's the bottom line purpose of the respiratory system, is to get that oxygen into the blood. The blood then carries it to the cells, and then the cells use that to make enough ATP to keep them alive. Without oxygen, we would die in about 5 minutes because the cells would be unable to meet our energy demands. Okay? So let's look at the anatomy of the nasal cavity. Not only is it there to warm and humidify and filter the air, but it's also there to provide us a sense of smell. There are olfactory receptors that are located on the mucosa of the superior surface. The rest of the cavity is lined with respiratory mucus, which helps moisten the air and trap the incoming particles. The lateral walls have projections that are called conchae that increase the surface area, so this allows the air that you breathe into the nasal cavity to have more contact with these membranes, and it also increases air turbulence within the nasal cavity. The nasal cavity is separated from the oral cavity by the hard palate. The anterior hard palate is made up of the maxillae that meet at midline, and the posterior hard palate is made up of the palatine bones, and then behind them is the posterior soft palate, which is essentially epithelial tissue, mucus secreting tissue, and then deep to that, muscle. And here you can see the paranasal sinuses. This is a series of hollow chambers that is continuous with the nasal cavity, and their purpose is to lighten the bones of the skull so that it's easier to use them. Okay, they're not so heavy, and it also has a role in humidifying, filtering, and warming the air that we breathe. So sinuses are located in the following bones: the frontal bone, the sphenoid bone, the ethmoid bone, the maxilla. They lighten the skull. They act also as resonance chambers for speech, and they produce a mucus that drains into the nasal cavity. And if you want to understand their role in speech, try to hold your nose and talk, okay? And you'll see how important they are in producing sounds. You can do that now. Try to talk with your nose closed, and you'll see that it's difficult because we don't have that resonating chamber. Okay, as the air moves around inside these hollow spaces, it allows us to produce a greater volume of sound. Here you can see the three zones of the pharynx: the nasopharynx behind the nose, the oropharynx behind the mouth, and the laryngopharynx behind the larynx. Um, the oropharynx and the laryngopharynx are common passageways for air and food. The nasopharynx normally is only there for the air, but food can occasionally be refluxed into the nasopharynx. I'm sure that you've perhaps tried to make someone laugh while they're drinking a beverage and had the drink come out their nose, so occasionally that can occur. It's important that the hard and soft palate separate the oral cavity from the nasal cavity for kind of this very reason. If these were continuous passageways, what would happen is it would be very easy for food and water to get into the nasal cavity and as a result end up in the airway. And there's a condition in which this happens called cleft palate, where the maxillae and or the palatine bones do not form a seal at midline, and a hole remains, and this makes the nasal cavity and the oral cavity a contiguous cavity. The problem is that in children it produces an inability to nurse. Okay, so this has to be corrected with craniofacial surgery. This problem can present in as minor a condition as a tiny defect at the front of the maxilla known as hair lip, or it can be as severe as a single open area for both the nose and the mouth. And we think that it may be linked to a deficiency of B vitamins in the diet of the mother, although that evidence isn't necessarily conclusive. The pharyngeotympanic tubes open into the nasopharynx. Another name for those are the eustachian tubes. The tonsils of the pharynx guard the oral cavity and act as a method of intercepting pathogens. The pharyngeal tonsil is located in the nasopharynx, the palatines are located in the oropharynx, and the lingual tonsils are found at the base of the tongue. And their purpose is simply to intercept pathogens. They are populated with macrophages and lymphocytes. The macrophage's job is to phagocytize the incoming pathogen, chop it into little bits, and then present those fragments on the cell surface along with major histocompatibility proteins as a warning to the lymphatic system that there is a pathogen that has gained access to the body. And then what will happen is that a team of B and T lymphocytes will be generated that will actually go out and fight the pathogen, knock it back. So we see here on the part of the body an excellent strategy, which is to put lymphatic tissue where it's most likely to encounter the pathogen. So here you can see the palatine tonsils, the lingual tonsils, and the pharyngeal tonsil up here. So you got a total of 5 tonsils. Now sometimes what can happen is that the tonsils can lose the battle with the pathogen and become infected and necrotic, at which point we have to remove the tonsils in a procedure known as a tonsillectomy. And you can live without your tonsils. There's plenty of other lymphatic tissue that can guard you against infection, even if you're missing some of these tonsils. And again, it's for the greater good. If those tonsils were to remain, you could see systemic infection and sepsis set in relatively short order. The larynx, or voice box, routes air and food to the proper channels and is important in speech because it contains the vocal cords. It's made up of 8 rigid hyaline cartilage and a spoon-shaped flap known as the epiglottis, and you can see it here. Okay, this is the larynx. Okay, there is the epiglottis, the thyroid cartilage shown right here. Okay, on men there's a bump on the front called the Adam's apple. Beneath that is the chrysoid cartilage. Whipping around to the back, you can see the rest of the chrysoid cartilage. There, of course, is the epiglottis. Those are the vocal cords. And then below the larynx begins the trachea, and you can see the tracheal cartilages forming C-shaped rings that terminate at the back of the pipe in order to allow the esophagus a chance to expand when you swallow food or water. So, the epiglottis is important because it protects the superior opening of the larynx and it routes food to the esophagus and air towards the trachea when you swallow. The epiglottis rises and it forms a lid over the opening of the larynx known as the glottis. The vocal folds contain what we know as the true vocal cords. They vibrate when expelled air causes them to oscillate, and that's what produces speech. The glottis is the opening between the vocal cords. And if you want to demonstrate to yourself how this operates, what you can do is you can get a can with an opening or a bottle, and you can blow air across the opening, and it'll cause the opening of the bottle or the can to shake, and you'll produce a sound. I don't have a good can here. I will endeavor to find a bottle for you, demonstrate it in class. If I had one with me, I could show you in short order. But anyway, I'm sure that you've done this with a Coke bottle, or some other beverage bottle in the past, blown across the surface and produced that low-pitched vibrating sound. That's how the chords work. The faster the chords vibrate, that generally reflects a greater tension in the cords. The cords have their tension adjusted by muscles that cause cartilages at the back of the cords to pivot. As those cartilages pivot and produce higher tension, that produces a higher pitch in the sound of your voice. As those cartilages pivot in the opposite direction, they reduce the tension in the cords, and that causes the pitch of the sound to be much lower. And what you're looking at here is just the trachea, which is below the larynx. It is a 4-inch long tube that connects the larynx with the primary bronchi. The walls are reinforced with the C-shaped hyaline cartilages that keep it open when you breathe in and out so the airway doesn't collapse. And it's lined with pseudostratified ciliated columnar epithelium, which is also populated with goblet cells that produce mucus. They beat continuously, moving the mucus up towards the oral cavity so that you can either spit it out or swallow it. And of course, the purpose of the mucus is to trap particulates, including pathogens, and keep them out of the lungs. And this avoids damaging the delicate lung tissue. When we get to the lungs, you'll see that these tissues are extremely delicate, easy to damage, because they are simple squamous epithelia and basement membrane, and as a result are very prone to scarring. You can see here, this is a cross section through the upper respiratory just below the trachea, or I'm sorry, just below the larynx. You can see here the lumen of the trachea. This is the pseudostratified ciliated columnar epithelium, so this is the mucosa. Below it is the submucosa, which is populated with mucus glands, and you can see cilia on the surface of the mucosa, and they are what do the beating, and they use ATP as an energy source. And then this is the cartilage that keeps the trachea open. This is a transverse section through the trachea, remember. And then notice that they end at the back of the trachea, and behind it is the esophagus, the food tube. And so you can see the purpose, again, of the C-shaped cartilages, which is to allow this pipe to expand when you swallow. And this is just a surface layer of connective tissue called adventitia, which is there essentially to glue the trachea to the surrounding tissue. So it's essentially a supportive function. And you can see here EM micrograph of the mucosa. You can see the cilia here, and you can see the epithelial and the goblet cells peppered all throughout. And these cilia constantly beat. Now occasionally you'll run into individuals that have a problem with these cilia. They may be, for instance, heavy smokers or exposed to high amounts of carbon monoxide. And what this does is it poisons the epithelial cells so that they can't beat. If this happens, the mucus that collects the particulates won't be moved towards the oral cavity. It'll end up in the lungs, and it'll damage the alveoli, causing them to scar and disappear. And this is one of the conditions that we associate very strongly with a disease known as emphysema. Okay. All right, the primary bronchi are formed by the division of the trachea. They enter the lung at an indentation called the hilum, which is just lateral to the heart. Okay, you can see a hilum here and a hilum here in this transverse section through the chest. This is the anterior, this is the posterior portion of the chest. You can see the heart and the great vessels. There is the right lung here, and this is the left lung, so that you know what you're looking at. The right bronchus is wider, shorter, and straighter than the left, and we should also note that the right lung has more lobes and more respiratory capacity than the left, and the reason for that is that the heart takes up room on the left side of the chest. Um, as a result, the right lung actually performs the bulk of the ventilation. The left lung is there simply to assist. This is also why sometimes when you choke, food particles tend to lodge in the right bronchus more readily than in the left. The bronchi will subdivide into smaller and smaller branches, and this is essentially a pipe that's designed to get the air down into the alveoli where the gas exchange can actually happen. So if we were to make a cast of the trachea and the bronchi and the bronchioles, and digest away all the soft tissue of the lungs and the upper respiratory and turn that cast upside down, it will look just like a tree, okay? And that's what we call the tracheobronchial tree. And it's very important because without this pipe, we wouldn't be able to effect gas exchange. Okay, now let's talk a little bit about the coverings of the lungs. The lungs occupy most of the thoracic cavity, while the heart occupies the mediastinum, which is the central portion here. The apex of the heart is near the clavicle. The apex of the lungs are near the clavicle. This is the superior portion, and the base rests right on top of the diaphragm, which is the main muscle of breathing. It is a broad, flat skeletal muscle. Each lung is divided into lobes by fissures, the left lung having 2 lobes and the right lung having 3. And it's very important to note, while we're looking here at the chest cavity, that the chest cavity itself is a closed cavity, okay? It is bounded by the diaphragm and the rib cage and the intercostal muscles, and in the back, and the sternum in the front, and in the back by the vertebral column and the muscles between the vertebrae. So this is basically a sealed chamber, and of course the lining of the inside of the thoracic cavity consists of the pleural membranes, which surround the lungs, and the pericardium, which surrounds the heart. And so the amount of air, okay, the total amount of air inside the thoracic cavity does not change during normal breathing. What does change is the volume that that air occupies, and that volume, the change in volume, is controlled primarily by the diaphragm, the main muscle of breathing, the intercostal muscles, the pectoralis major and minor, and to some extent the sternocleidomastoid, and then at the back, the erector spinae. Okay, and we'll talk a little bit about the mechanics of breathing in a bit. But remember that these lungs exist inside an air-filled chamber, but the amount of air in that chamber does not change. The volume of the chamber does, however. Okay. The serosa covers the outer surface of the lungs. The pulmonary pleura covers the lung surface, while the parietal pleura covers the chamber inside which the lung sits. And between the pleura is a pleural fluid, which is a lubricant. And what this does is this eases the movement of the lungs as you inhale and exhale, reducing irritation, inflammation, and friction, which would otherwise damage the delicate tissue. The 2 pleural layers resist being pulled apart. They kind of cling together a bit, and this is important because when the volume of the chest cavity changes, what happens is that we produce now, when the diaphragm is contracting, a vacuum around the outside of the lungs, which allows them, as they cling to the thoracic cavity, to expand. And then when we breathe out and the diaphragm rises, that increases the pressure inside the thoracic cavity, and what that'll do is cause the intrapleural pressure to go up, forcing the air in the lungs out. Okay, so that's just a little brief introduction to the mechanics of ventilation, but it wouldn't be possible if the lungs didn't cling to the walls of the thoracic cavity when you breathe in and out. And we'll demonstrate in lab negative pressure breathing with a model that I'll show you. The divisions of the respiratory tree are basically formed by ever tinier branches that come off the trachea, forming then the primary bronchi, then the secondary bronchi, and they continue to branch and branch and branch until we get to what we call the terminal bronchioles. And these are very tiny structures inside the lungs which do not engage in gas exchange. They're simply there to conduct the air to the lungs where the gas exchange will actually take place. Okay? Now, what happens next after the terminal bronchioles is that we see now the respiratory bronchioles that lead into alveolar ducts that eventually lead into alveolar sacs, and this is where the gas exchange takes place. And this is really the money part of respiration, because this is where we swap oxygen for carbon dioxide. The carbon dioxide gets blown off, the oxygen gets loaded in to the circulatory system, and that oxygen-rich blood then flows back to the heart, which then pumps it out to the rest of the body. Notice that as we look at the alveolar ducts and the alveolar sacs, that they're very thin. Okay, the linings are very thin. They're also slightly moist. There's a very special secretion called surfactant, which is sort of like a detergent that overcomes the surface tension of water. This allows the lungs to very easily expand and contract when you breathe, so that it's easier to ventilate the lungs. Now, on the other side of the alveoli are lots of capillaries, and so the maximum distance that the gas has to travel is about 2 cell layers thick, and the tissue that the alveoli are made out of is simple squamous epithelia, very, very narrow tissue, and of course the capillaries, as you already know, are made out of endothelial tissue and basement membrane. So basically, what we call the respiratory membrane consists simply of air that flows out of the alveoli and into the capillaries, and only has to cross 2 cell layer thicknesses and 1 shared basement membrane. So there's not much of an impediment to gas movement in normal, healthy, functioning lungs. Now, sometimes we can damage this tissue, and if we do, we can impede the exchange of gases. For instance, in pneumonia, where we start to have fluid collect in the lungs, or in the case of pleural effusions, that liquid provides a barrier to gas exchange that impedes the loading of oxygen into the circulatory system and is going to basically diminish the ability of the lungs to deliver oxygen to the tissues. As a result, there will be less ATP available. As a result, we'll tend towards easy exhaustion. And if this keeps up too long, eventually what can happen is we can start to see tissue death. So any condition that thickens this layer is bad. Emphysema, which we already talked about, affecting the mucosa of the upper respiratory, also damages alveoli, causing them to scar and also to be coated with tar that comes from cigarette smoke. And the danger, of course, with this is that scar tissue is thicker than epithelial tissue. It impedes gas exchange, and ultimately the result is diminished ability to exchange oxygen for carbon dioxide. Another example is black lung disease or silicosis, diseases where particulates get inhaled into the lungs, causing scarring, damaging the alveolar tissue, and impeding gas exchange. And then finally, one of the most insidious conditions is one that's called cystic fibrosis, or CF, which is a congenital disorder. It's basically a defect in a pump that is tasked with maintaining electrolyte concentrations inside and outside all the cells in the body. The effect that this has on the surfactant that's produced in the lungs of the respiratory system is that it causes it to be too thick, and as a result it provides a barrier to gas exchange, and it also irritates and inflames the lungs, causing them to scar. And so really you have a double problem here. Not only is the mucus a barrier, but the scar tissue is a barrier as well. There's no cure for cystic fibrosis. There are treatments in the infant or in the child what we can do is hold the child upside down and percuss the back and allow the mucus to be coughed up with the assistance of an inhaler that contains DNA, which breaks up the DNA that's released into the mucus as the bacteria that grow in it die and release their cellular contents. So it helps to thin the mucus out. In the adult, we can't do that. We have to have a device, and the device is called an ambivest. And what the ambivest does is it plugs into a power supply and it beats very vigorously. And as the infected mucus is produced in the lungs, what the ambivest will do is allow the CF patient to cough it up so that the lungs remain clear, and this will assist with gas exchange. So again, it's not something we can fix. It is something, however, that we can screen for. What you're looking at here is just the respiratory division of the respiratory system. You can see here the respiratory bronchioles leading into the alveolar ducts, which lead into the alveolar sacs. This is the same diagram from before, but here you can see an electron micrograph of alveolar tissue, and it has the exact same consistency as a wet sponge. It is that filled with these air-filled chambers. You would not know if you were actually to see preserved lung tissue. You would think that it was styrofoam, literally. But that is, in fact, what lung tissue is like. Okay, so the alveoli are the air sacs, and they are where the gas exchange actually takes place. And what you're looking at here is just the respiratory membrane that I was telling you about. The thin squamous epithelial layer lines the alveolar walls. The alveolar pores connect to the neighboring air sacs, and pulmonary capillaries cover the external surfaces of the alveoli. On one side of the membrane is air, and on the other side is blood flowing past. And you can see in the diagram, this is an alveolus, okay? And you can see that it's dominated by simple squamous epithelium. We'll talk in a second about the other cells that are in there. But look here at the respiratory membrane. This is the alveolar chamber that's filled with the air that you inhale. And look at how short the distance is that the oxygen has to traverse to get into the blood, and the carbon dioxide at the same time has to move across to get into the alveolar air, which is then blown off. Okay, so you can see there the alveolar epithelium, the fused basement membranes, and of course the capillary endothelium has to remain thin like that in order for the gas exchange to happen efficiently. Now there's other cells in here besides the simple squamous epithelial cells. You'll notice that there are occasional green cells in here. These are called type 2 alveolar cells, and what they do is secrete surfactant, and that's the detergent-like secretion that allows the lungs to very easily expand and contract when you breathe. Okay, so it overcomes that surface tension. And then there's also a lot of macrophages in the lungs, and their purpose is to simply intercept pathogens and kill and recycle dead tissue and keep the lungs free from infection. And you'll also note that around the lungs are a lot of lymph nodes and lymphatic capillaries, and what they do is anything that gets past these macrophages will end up in the lymphatic system, where it'll eventually be escorted to a lymph node, which will alert the lymphatic system to the presence of the pathogen, and then will draft our army of lymphocytes that will go out now and fight and destroy this pathogen. Now, gas crosses the respiratory membrane by diffusion. The oxygen enters the blood, and the carbon dioxide enters the alveoli, while the macrophages are there to protect against bacteria and particulates. The surfactant coats the gas-exposed alveolar surfaces, and that's what we're talking about, this detergent that overcomes the attraction of water for itself inside the lungs. Without surfactant, it would be very difficult for you to inflate the lungs. It would be like trying to inflate a wet paper sack that had all the air squeezed out of it. You know how hard that would be. And if you want to do an experiment, try inflating a wet paper sack, and then try inflating a paper sack that's been moistened with soapy water, and see how much easier that is. Okay, and that's again because the detergent effect of the water overcomes the attraction of the water for itself. So there's four events in respiration that we need to understand. Pulmonary ventilation is simply the moving of the air in and out of the lungs, also known as breathing. External respiration is the gas exchange that occurs between the pulmonary blood and the alveoli. Here the oxygen is loaded into the blood and the carbon dioxide is unloaded. Respiratory gas transport is the transport of oxygen and CO2 by the blood. And internal respiration is gas exchange between the blood and the tissue cells in the systemic capillaries, those that provide blood and gas exchange to the rest of the body. So if we look at the mechanics of breathing, essentially what we're examining is how is it that the air gets in and out of the lungs. And the bottom line is this happens primarily by muscle contraction. It's a mechanical process that depends on volume changes of the thoracic cavity. Volume changes will lead to pressure changes, which will lead to the flow of gases to equalize pressure. There's two phases: inspiration, which we know as inhalation, where the air flows into the lungs, and expiration, known as exhalation, where the air leaves the lungs. And so what you're looking at here is a figure that shows you the diaphragm with the heart and lungs inside, and the thoracic cavity with the heart and lungs inside, and the diaphragm at the inferior border. And what I want you to notice is that it's a closed chamber, okay? And so the amount of gas in that chamber does not change. The diaphragm and the external intercostal muscles contract, and the size of the thoracic cavity increases when you inspire, when you take air in. The external air is pulled into the lungs due to the increase in intrapulmonary volume and the decrease in the gas pressure. Essentially, the reason that we call this negative pressure breathing is because a negative pressure is defined as a vacuum. So when the diaphragm contracts, what it does is it causes the thoracic cavity to enlarge. That produces a negative pressure, a vacuum around the outside of the lungs. The lungs will cling to the pleura, okay, the parietal pleura, and that vacuum will then be translated to the lungs themselves. And if your nose or your mouth are open, the air will rush into the lungs. And so what you're looking at here is a chart showing you the changes in intrapulmonary pressure when you inhale and when you exhale. Okay, so what you're looking at on the y-axis here is the pressure relative to atmospheric pressure. So when you are inhaling, notice that what you produce is a vacuum, a negative pressure inside the chest cavity. And when you blow out, when you exhale, what happens is you produce a positive pressure inside the chest cavity. And if the nose and the mouth are open, the air will always flow from an area of high pressure to an area of low pressure, and that's also true for liquids. And here you can see the volume of the breath that corresponds to this inhalation and exhalation. Okay, so here we are at the beginning of inspiration. Okay, we breathe in. Okay, the volume goes up, 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 and then we breathe out, and the volume goes down, 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 down. Okay, and here you can see a transverse section through the thoracic cavity. Notice that at full inspiration, with the external intercostals and the diaphragm contracting, that we have a lot of extra space inside the chest cavity, and that's what produces the vacuum around the lungs that allows us to breathe. Now with expiration, we simply reverse the process. What happens is that the intercostals and the diaphragm, the pecs, the sternocleidomastoid, they all relax. The diaphragm goes from a flattened to a domed shape. The volume of the thoracic cavity reduces dramatically. This produces a positive pressure outside the lungs. Okay, you can see that here. And what happens now is that the breath is expired. Okay, this is essentially a passive process. Okay, you don't need to use muscular contraction in order to drive the air out of the lungs, but you can. There are muscles that are used in forced expiration, but those are primarily the muscles of the abdominal pelvic cavity, the rectus abdominis, the transverse, the internal and external obliques. And when they contract, what they do is they push the internal organs up against the diaphragm. This further decreases the volume of the chest cavity, and this forces more air out of the lungs. Okay. In forced inspiration, we use more of the intercostals and the pecs and the sternocleidomastoid to expand the volume of the chest cage in an effort to help the diaphragm pull air into the lungs. So that's forced inspiration. Okay, normal pressure in the pleural space is always negative. This is what we call intrapleural pressure. The differences in lung and pleural space pressures result in the lungs not collapsing. Non-respiratory gas movements can be caused by reflexes or voluntary actions, and examples include coughing and sneezing to clear out debris from the lungs, crying, which is an emotionally induced mechanism, and laughing, and also hiccups, which are sudden inspirations, and yawns, which are very deep inspirations. Now, when we look at the ability of the muscles of breathing to affect ventilation and the elasticity of the lungs in assisting ventilation, we have to have a tool in order to gauge whether or not your respiratory system is doing what it's supposed to, and this tool is called a spirometer. And what a spirometer is essentially, in the old style spirometers, it's a cylinder within a cylinder. One cylinder contains water. A second cylinder, which is smaller than the first, floats on top of this water, and there's a pipe that blows air underneath the second cylinder and causes it to rise and fall as you breathe. And that drum, as it moves up and down, is attached to a pen which will draw on a chart on a third drum that rotates. Okay, so that as the drum rotates and as the pen goes up and down, you can determine the volume of air that moves in and out of the lungs in different breathing actions. Okay, so if we look here at a typical spirometer tracing, what you're seeing here is the pen actually writing on the paper that's attached to the drum of the spirometer. Okay, when you see a peak going up, that represents you breathing in, and when you see a trough appearing, that represents you blowing out. Okay, so let's draw that. Okay, this is okay, breathing in. Okay, and down here this is breathing out. Okay, so normal breathing, as you're just sitting there breathing quietly, moves about half a liter of air in and out of the lungs with each breath. This is what we define as the tidal volume, and so the tidal volume is indicated here by this light blue bar. Okay, you can see 500 mils as the amount of air that you move in and out in a normal breath. Many factors will affect this capacity. They include a person's size, his sex, his age, and his physical condition. Other volumes that we measure using a spirometer include the vital capacity. Okay, now what is the vital capacity? That's the maximum of air that you can breathe in and then blow out using all the muscles you can, and that's represented by this arrow that I'm drawing here. It's from the peak of this bump here to the end of this trough. Okay, that's what we call our vital capacity. Okay, this we've also already indicated is your tidal volume from here to here. That's your normal breathing. And then you've got a few other capacities. Okay, if you were to breathe in normally, okay, and then stop and don't breathe out, and then continue to breathe in, okay, that amount of air that you could pull into the lungs after you continue to breathe in is what we call your inspiratory reserve. Okay, and that's indicated here by this yellow arrow. Okay, this yellow arrow. By the same token, if you were to breathe out normally and then stop and don't breathe in again and then breathe out as hard as you can, that would be what we call your expiratory reserve, and that would be indicated here by this green arrow right there. I know it's a little hard to see, but bear with me. Okay. So you've probably guessed that there's a mathematical relationship to these different volumes. Your vital capacity is equal to your inspiratory reserve plus your tidal volume plus your expiratory reserve. Okay. Now, there's an amount of air that you can't move out of the lungs no matter how hard you try, and that's called your residual volume, and that's there essentially to keep the lungs to maintain their shape. And it's not air that's available for gas exchange. So if you were to do a vital capacity, to breathe in as hard as you can and out as hard as you can, after the end of your vital capacity, okay, the bottom of this red arrow right here, there would still be some air inside the lungs, and that is your residual volume, and we'll indicate that with a white arrow right here, okay? And there's really no direct way to measure that short of removing your lungs from your chest cavity, pushing all the air out and collecting it in a bottle, okay, and then measuring that volume. So your total lung capacity is equal to your residual volume plus your vital capacity. So that's the white arrow here plus this long red arrow. All right. And again, we can't directly measure residual volume. Now, what's going to affect these lung volumes and capacities? Well, the elasticity of the lungs, how well they stretch and contract, the strength of the muscles of breathing, okay, and whether or not there are any secretions inside the lungs that might impede their movement, okay, such as pleural effusions or CF-style mucus or anything like that. All right. So this is just an explanation of these different volumes, right? Inspiratory reserve, the amount of air that can be taken in forcibly over tidal volume. It's usually around 3.1 liters. The expiratory reserve, the amount of air that can be forcibly exhaled, that's about 1.2 liters. The residual volume, the amount of air remaining in the lungs after you blow out, and that's about 1.2 liters tops. Okay? So let's examine some of these respiratory capacities in a little more detail. The vital capacity is the total amount of exchangeable air that you can move in and out of the lungs with maximum effort, and it's defined as the tidal volume plus the inspiratory plus the expiratory reserve volume. Dead space volume is the air that remains in the conducting zone and never reaches the alveoli, and this is what we call the part of the residual volume, and it's about 150 milliliters, while the functional volume is the air that actually reaches the respiratory zone, and on a normal breath that's about 350 milliliters. So you could think of essentially this volume here as representing what fills up the trachea, the bronchi, and the bronchioles up to the terminal bronchi, and also what helps to maintain some of the shape of the lungs. While this volume down here, the 350 milliliters, is the part that's actually in contact with alveolar membranes and is capable of gas exchange. So you can see that every time you perform a normal tidal volume, that approximately a fourth of what you breathe in is unavailable for gas exchange. Respiratory capacities, as we already pointed out, are measured with a spirometer, which is a device that you blow into, which is attached to a needle, which draws on a rotating drum that is covered with chart paper. And I'll show you an example of an operating spirometer at the end of this presentation, so you have an idea how the machine operates. But it's essentially a device that's going to measure the strength of your muscles of breathing along with the elasticity of your lungs. Now, respiratory sounds are monitored with a stethoscope, and there's two recognizable sounds that you can hear: bronchial sounds, which are made by air rushing through large passageways like the trachea and the bronchi, and breathing sounds, which are the soft sounds of the air filling the alveoli. If you hear other sounds, such as wheezing or rales, that indicates some sort of buildup in the conducting division, which can impede the movement of air. And it's generally an indication of perhaps an infection of the respiratory or some other type of obstructive disorder in the respiratory system, basically a block to the air getting in and out of the lungs. Oxygen that's loaded into the blood at the alveoli diffuses down its concentration gradient from the alveolar air directly into the red blood cell. The alveoli are always going to have more oxygen than the blood that's coming to the lungs from the heart, because the amount of oxygen in the air that you breathe is 20% by volume, and it's considerably less in the oxygen poor blood that's perfusing the capillaries that are on the other side of the respiratory membrane from the alveolar air. So the oxygen will move by diffusion where it's more where the concentration is lower, and that's something that happens automatically without needing to put energy into the system. The pulmonary capillary blood picks up oxygen as a result and will transition from a dull red to a bright red, whereupon it will also simultaneously unload its carbon dioxide, and then that oxygen-rich blood will return to the lungs through the pulmonary veins. The carbon dioxide is unloaded out of the blood because its concentration gradient is the reverse of the concentration gradient of the oxygen. The amount of carbon dioxide in the air that you breathe is less than 1%, where it's considerably higher in the oxygen-poor blood that's coming to the lungs from the heart. So the blood returning to the tissues after it passes through the lungs is going to have a higher concentration. I'm sorry, that's returning to the lungs from the heart is going to have a higher concentration of carbon dioxide than the air in the alveoli. And this is why you can see in this diagram the carbon dioxide diffusing out of the blood while the oxygen simultaneously diffuses in. Pulmonary capillary blood gives up carbon dioxide that's going to be blown off when you exhale, and the blood leaving the lungs as a result is going to be oxygen rich and carbon dioxide poor. That's why it has that bright red color as it returns back to the heart through the pulmonary veins. Most oxygen travels in the blood attached to hemoglobin, forming molecules of oxyhemoglobin, indicated here as HbO2. A small amount is carried in the plasma, but not really very much. Carbon dioxide, on the other hand, is transported primarily in the plasma as bicarbonate, and a small amount is carried inside red blood cells attached to hemoglobin at binding sites that are different from where the oxygen binds to the hemoglobin. And it's a curious property of carbon dioxide that as carbon dioxide binds to hemoglobin, it tends to release its oxygen, and as oxygen binds to hemoglobin, it tends to release its carbon dioxide. And again, that's to our advantage, because that means that hemoglobin will tend to bind oxygen in the lungs and will tend to release oxygen when it gets to the tissues. For carbon dioxide to diffuse out of the blood into the alveoli, it has to be released from its bicarbonate form. What happens is that the bicarbonate ions enter the red blood cell, combining with hydrogen ions to form carbonic acid, which then, under the influence of an enzyme called carbonic anhydrase, will split to form water and the gas carbon dioxide, which diffuses into the alveoli. And you can see that process here, okay, bicarbonate combining with protons making carbonic acid, and in the red blood cell, this will quickly be converted into carbon dioxide and water. It'll also dissociate in free plasma. So you can see the carbon dioxide again diffusing into the alveoli. Now, internal respiration is defined as the exchange of gases between the blood and the body cells. An opposite reaction to what occurs in the lungs is what takes place in the tissues. Carbon dioxide diffuses out of tissues and loads into the blood basically converting into carbonic acid, which becomes bicarbonate, while at the same time some of that carbon dioxide diffuses into the red blood cell, binding to hemoglobin and causing it to let go of its oxygen. So you can see here the oxygen diffusing out of the red blood cell and into the tissues. Where does the carbon dioxide come from? Well, the carbon dioxide comes from metabolism. So let's use blue for carbon dioxide. Okay, and this is from metabolism. MET, metabolism. Okay, and it diffuses into the blood, while the oxygen here, we'll indicate that with a red arrow, comes from air. Okay, so what happens at the tissues is that oxygen leaves the hemoglobin, carbon dioxide binds to the hemoglobin, and also carbon dioxide leaves the tissues and is carried in the plasma as bicarbonate and also by the hemoglobin as carbaminohemoglobin. And so this oxygen poor blood will return eventually to the heart, which will pump it to the lungs, and there we'll repeat the cycle of oxygen loading and carbon dioxide unloading and repeat the entire process. And what you're looking at here is just sort of a diagram showing you how oxygen and CO2 levels change as we move through the circulatory system. So we can begin here with blood leaving the tissues and entering the lungs, where you can see the carbon dioxide levels are higher than the oxygen levels. And then what happens is that the oxygen poor blood gets pumped into the lungs. At the alveoli, we have loading of oxygen into the blood and unloading of carbon dioxide. So you can see here how much oxygen there is in inspired air compared to CO2, which is virtually non-existent. And then in the alveoli of the lungs, there's still quite a bit of oxygen compared to carbon dioxide in alveolar air. And what this does is this tends to force oxygen into the blood, and carbon dioxide is coming out of the blood. And so that's why you're starting to see these carbon dioxide levels rise a bit. What happens by the time that blood leaves the lungs is that its oxygen content is very high compared to when it went in, and it still has some carbon dioxide content, but not nearly the amount that it had when it was originally coming to the lungs from the heart. What happens now is that as we return to the heart, that oxygen-rich blood is going to be pumped out of the left ventricle into the aorta, and from the aorta we're going to basically be moved through the entire body. And what will happen when we get to the active tissues is that we'll see an unloading of oxygen into the tissues and a loading of carbon dioxide. Notice that in the tissues we have a high concentration of carbon dioxide because that's an end product of metabolism, and a low concentration of oxygen because oxygen is constantly being consumed in metabolism as a method to produce ATP that the cells need in order to grow and repair and divide. And then of course the oxygen poor blood returns to the heart and the entire cycle repeats itself. Now the activity of the respiratory muscles is transmitted to and from the brain by the phrenic and the intercostal nerves. The phrenic nerves talk to the diaphragm, and the intercostals talk to the muscles that are between the ribs. There are also neural centers that control the rate and depth of breathing that are found in the medulla and the pons, which remember are part of the brain stem. The medulla sets the basic rhythm of breathing and has pacemaker cells that are self-exciting, and this is what we call the inspiratory center of the brain stem, while the pons appears to smooth out the respiratory rate. So it's a modulator of breathing rhythm. The normal respiratory rate is between 12 and 15 respirations per minute, and this is termed eupnea, E-U-P-N-E-A. While hyperpnea is an increase in the respiratory rate, often due to increased oxygen need, say under situations of strenuous exercise or of imminent panic. And the idea here is that in those situations, the body needs more ATP, so we need a higher respiratory rate and heart rate in order to deliver sufficient oxygen and nutrients to the tissues to fill their energy needs. And so this is why the respiratory rate increases. And so what you're looking at here is just how the interplay of these systems work. You can see that, again, the brain is bathed in cerebrospinal fluid, and the changes in carbon dioxide signal the brainstem to cause the breathing center to fire. Okay, these afferent impulses hit the medulla. The medulla, in turn, is going to innervate the phrenic and the intercostal muscles. You can see here and here, and that's going to accelerate the breathing rate. Okay, and again, this will be modulated by influences from the pons, that part of the brainstem that sits right above the medulla oblongata. Okay. Other stimuli that can trigger an increase in breathing rate comes from oxygen sensors that are in the aorta and the carotid bodies that are in the carotid arteries that sense a drop in oxygen. They too can fire afferent impulses to the medulla, which in turn is going to accelerate the breathing rate in order to increase the amount of oxygen loaded into the blood per unit time, and as a result pay our oxygen and as a result our energy debt. So there are also non-neural factors that influence the rate and the depth of breathing, and these include increased body temperature, exercise and talking, coughing, and there's also conscious control over respiratory rate. This is called acts of volition, which include emotional factors and also just simple conscious modulation of breathing. Chemical factors also have a major influence over respiratory rate. The body's need to get rid of carbon dioxide is the most important stimulus. As carbon dioxide levels in the blood rise, the respiratory rate goes up, and also as the pH of the tissue fluid and the plasma go down, the respiratory rate goes up. And the reason for this is that carbon dioxide levels have a direct effect on the pH. Remember that carbon dioxide, as it accumulates in the plasma, forms carbonic acid, which in turn will dissociate, forming protons that will cause the pH to drop. And this in turn will eventually cause the rate of breathing and the depth of breathing to increase. The changes in CO2 concentrations act directly on the medulla. Oxygen is also an important stimulant. Changes in oxygen concentration in the blood are detected by carotid and aortic sensors, and their information is sent to the medulla. The lower the oxygen content, the greater the rate and the depth of breathing. The higher the oxygen content, the more we're going to tend towards normal rates of breathing, both in depth and in rate. Hyperventilation results from increased carbon dioxide accumulation in the blood, which can lead to acidosis. The breathing becomes deeper and more rapid in an effort to blow off the carbon dioxide, and when this happens, the protons are going to be consumed in the plasma, forming a carbonic acid, which is going to then dissociate into CO2 and water, and of course the CO2 is going to leave with the expired breath. So this is one of the ways that we can directly control the body's pH. In hypoventilation, the blood becomes alkaline. This causes the rate and the depth of breathing to slow down, causing carbon dioxide to build up in the tissues, and this causes the pH to begin to drop, which is exactly what we want in this case. Now, there are different disorders that can affect the respiratory system's ability to ventilate the lungs. And the two broad categories are what we call restrictive disorders and obstructive disorders. And let's clarify the difference between the two. An obstructive disorder is basically a block in the tracheobronchial tree that cuts down on airspeed into and out of the lungs. Examples include things like asthma, um, the common cold, okay? Restrictive disorders are conditions in which the lungs are unable to expand to their full volume, either because they've lost elasticity or because the dimensions of the thoracic cavity have been altered, just to name a few. An example of such a disorder might be emphysema, where the lungs will scar and as a result become less elastic, and also scoliosis, where the volume of the thoracic cavity is altered due to a sideways curvature of the spine. So those are just a few examples. Chronic obstructive pulmonary disease is essentially a condition that cuts down on airspeed, and it's exemplified by chronic bronchitis and emphysema, and it's a major cause of death and disability in the U.S. The patients almost always have a history of smoking, and they exhibit labored breathing, which becomes progressively more severe. And the reason for this is that the lung tissue is accumulating tar, which is killing alveoli and producing scar tissue, and also cutting down on the ability to exchange oxygen for carbon dioxide in the lungs due to a thickening of the respiratory membrane. The scar tissue thickens it, and the tar, as it accumulates on the alveoli, thickens it as well. Features of these diseases include coughing and pulmonary infection. Also, we see hypoxia, a buildup of carbon dioxide in the plasma and in the tissue fluid, and often respiratory acidosis, because remember, as carbon dioxide levels increase, carbonic acid production increases, and that causes the pH to drop. Those that become infected as a result of COPD will often develop respiratory failure. Chronic bronchitis is more of a restrictive disorder. It's an inflammation of the bronchi. The passages become severely inflamed, mucus production increases, and it impairs ventilation and gas exchange. So what's happening is that the mucus is thickening the respiratory membrane, and it's also narrowing the passages of the bronchi and the trachea and letting less air get into the lungs. As a result, you have to breathe with increased effort. This is why very often people with chronic bronchitis can develop a barrel chest due to the overexertion of the respiratory muscles, the intercostals, the pecs, the diaphragm, and so on. The risk of lung infection goes up because of the large amount of mucus that's produced. Mucus serves as a fuel for the bacteria that are in the air that we breathe, and as they begin to grow, they cause the mucus to thicken, and that can inflame the lung tissue, causing it to scar and reduce its flexibility, and also the thicker mucus impedes gas exchange directly. Pneumonia is very common in these individuals, as we have fluid in the lungs due to the complications of this condition. They're often nicknamed blue bloaters because they exhibit both hypoxia and cyanosis. Emphysema is where the alveoli are destroyed due to tar that's introduced into the lungs, often due to cigarette smoking. What happens here is that the alveoli enlarge as chambers basically burst, and what we see now are fewer alveoli, and as a result, less area for gas exchange. The inflammation promotes fibrosis in the lungs. This is basically the lungs scarring, and the airways often collapse when we expire. When we breathe out, the trachea and the bronchi will basically close up on themselves. The patients use a large amount of energy to exhale, and this overinflation of the lungs will lead to a permanently expanded barrel chest, again because they have to work harder in order to get the air in and out of the lungs. Cyanosis usually appears late in the disease. Sufferers are often called pink puffers as opposed to blue bloaters. And so here you can see how it is that these different environmental components can impact the efficiency of the respiratory system. Tobacco smoke and air pollution contain carbon monoxide and also tar and particulates. This produces inflammation in the lungs, can lead to bronchitis, mucus production results. We have productive coughs. This is where you cough up lots of sputum. We tend to see bronchospasms, and this promotes airway obstruction, dyspnea, and frequent infection, and ultimately respiratory failure. It can also result in the breakdown of elastin in the connective tissue of the lungs, so they're less stretchy. The emphysema results in a destruction of valvular walls. The lungs become fibrotic, so they become basically scar tissue, and this results in air trapping because the lungs are unable to expand and contract as they used to during the course of normal breathing. Okay, again, scar tissue also is thicker than normal alveolar tissue, and it also results in less ability to load oxygen into the lungs, into the blood that comes to the lungs from the heart directly. Lung cancer accounts for a third of all deaths, all cancer deaths in the U.S. The increased incidence is associated with smoking cigarettes because cigarette smoke is inhaled into the lungs, whereas other types of smoking, normally cigar, pipe, and so on, are not inhaled into the lungs. So there's less possibility for tar to build up inside the alveoli and the bronchioles. There's three common types: squamous cell carcinoma, adenocarcinoma, and small cell carcinoma. And the bottom line with cancer is that uncontrolled cell growth leads from primary tumors to metastasis, that secondary tumors begin to set up in other locations in the body, such as the bone marrow and the brain. And eventually what happens is that this rapidly growing tissue removes so many oxygen and nutrients from the blood that it ends up killing the host. Now, if we look at the way the lungs form, it's very interesting because while you're in your mother's uterus, you don't really need your lungs in order to get oxygen. And this is because most of the oxygen comes into the embryo and the fetus through the blood that comes back to it from the umbilical cord. And the umbilical cord serves as an interface between the mother's blood and the baby's blood. So what happens when the blood is coming from the mother filled with oxygen and nutrients is that it comes into very close contact with the blood that's coming from the baby, and even though they don't mix, what happens is that we exchange oxygen and carbon dioxide and nutrients and waste products at the placenta, and then that oxygen and nutrient-rich blood flows back to the embryo or fetus, and eventually to all the tissues in the body, and that's what fuels growth. In the fetus, in utero, the lungs are filled with amniotic fluid, and they're not fully inflated until 2 weeks following birth. And what happens during that time is that surfactant production increases in the respiratory system until by the time the baby's born, we have adequate surfactant to support relaxed breathing. It's a detergent molecule that's made by the alveolar cells that breaks the surface tension of water, and this allows the lungs not to collapse so easily when we breathe. It's like a soapy secretion, and so it becomes very easy for the lungs to expand and contract when the water molecules aren't adhering to each other as strongly as they otherwise would. It's not present in significant amounts until late in fetal development, and in premature babies we often have to give exogenous surfactant through an inhaler in order for them to breathe properly, because again we don't have adequate production of this detergent component in the lungs so that breathing can occur without assistance. It generally appears in a normal pregnancy between 28 and 30 weeks. And obviously, by the time the baby's born, there's sufficient surfactant in the lungs that normal breathing can continue unabated. Sometimes what can happen is that if the baby's born too early, there's lack of surfactant, and this will result in an inability of the child to properly breathe, and this is called infant respiratory distress syndrome. And this is where we have to give surfactant through an inhaler in order for them to breathe without tremendous effort. Cystic fibrosis is an oversecretion of thick mucus that clogs the respiratory system, and it's the result of a defect in a sodium pump that's on the surface of every cell in the body. Results in the production of an extremely viscous mucus that gets populated by bacteria and becomes very difficult to clear out of the lungs. And this leads to scarring of the lungs and also the mucus itself impedes gas exchange. And so there's really no way to fix this problem. We can deal with some of the symptoms. We can thin the mucus using inhalers that contain enzymes that break apart DNA, because DNA is one of the main components that thickens the mucus in CF patients. And then we can also, in infants, percuss the lungs, beat on the back of the infant so that they can cough up the excess mucus so that they can continue to breathe for another day. But as obviously the child grows up, we have to use another approach in order to clear the mucus out of the lungs, and that's where the AmbiVest comes in, which is essentially a device that looks like a life jacket that plugs into a power supply and beats very rapidly along with the inhaler. That action allows the young adult or the adult to expel the mucus out of the lungs so that they can again continue to breathe relatively normally. Again, this isn't something that we can fix. It is something we can screen for, however, in amniocentesis, so we can prepare the parents for life dealing with the disease, and as a result, they'll be adequately prepared once the baby comes to at least make the early stages a lot more comfortable. Respiratory rates change throughout life. Newborns breathe between 40 and 80 respirations per minute because they are growing, and as a result they need more air in order to produce more ATP, in order to provide enough energy for all the changes that are going on in a growing baby. Whereas infants slow down to about 30 respirations per minute due again to the decreased amount of energy required for the infant. And then by age 5, we're down to 25 respirations per minute, and in the adults, between 12 and 18. These rates sometimes increase with old age because the strength of the respiratory muscles and the elasticity of the lungs decreases, and as a result, we often have to accelerate our rate of breathing in order to compensate for the diminished capacity of the lungs to fill with air. Your vital capacity basically drops the older you get, and also for the weakened state of the respiratory muscles, which can't allow the lungs to expand as fully as we would have experienced, say, in young adulthood. Sudden infant death syndrome is where apparently healthy infants stop breathing and die in their sleep. Some cases are thought to be a problem with the neural respiratory control center that's in the brain. About a third of cases are thought to be due to heart rhythm abnormality, but recent research shows that there may also be a genetic component. Essentially, when we are unconscious, it's the periodic stimulation of the phrenic nerve that allows the diaphragm to contract and relax, and this is what facilitates the ventilation of the lungs. And in a percentage of SIDS death, we believe that what happens is that periodic stimulation that normally takes over when we fall asleep fails for some reason, and as a result the diaphragm quits moving. The result is that ventilation of the lungs ceases, and this can result in death while sleeping. Asthma is a chronic, inflamed, hypersensitive condition of the bronchioles. Basically, what happens is that as they irritate, the lumen of the bronchioles tightens. It becomes narrower and narrower, and the response is usually dyspnea, coughing, and wheezing. If you've ever seen anybody have an asthma attack, um, it's um, it's a frightening thing because essentially they're suffocating because they can't get sufficient air into the lungs um, to maintain consciousness. And so asthmatics will often carry with them inhalers that contain epinephrine or albuterol, and what these compounds do is they promote bronchodilation so that you can more easily inflate the lungs when you breathe. As we age, the elasticity of the lungs declines and the vital capacity goes down because the lungs can't stretch out as much. The blood oxygen levels decrease and the stimulating effects of carbon dioxide decrease. As a result, the elderly are often hypoxic and will exhibit sleep apnea, and they also run a higher risk of respiratory tract infection. Just what you have to understand is that even if you've never smoked in your life, as a result of a lifetime of breathing, the air that you breathe is not perfectly clean. It's filled with particulates and pathogens, and the result of this over a lifetime is to damage the lungs, to cause them to scar, to cause them to lose their elasticity, and along with that, at the same time, all our muscles are becoming weaker as we age. And so this results really in a two-pronged attack on our vital capacity. Not only is it difficult for us to ventilate the lungs as effectively as we would like um, because they don't stretch out as much, but we also don't have the pull from the muscles of breathing, the strength from the muscles of breathing to produce a sufficient vacuum to get enough air into the lungs in order to support normal oxygen levels as we did when we were younger. And so that's going to invariably happen the older we get. The lungs are going to lose their ability to properly ventilate, and as a result, the ability of the respiratory system to load oxygen into the body is going to decline, and as a result, our energy and activity levels will decline as well. Okay, stay tuned for a little explanation of what a spirometer is and some discussions of respiratory volumes and the movement of gases in the respiratory system. And I'll see everybody in class.
Lung volumes and capacities, with the exception of the residual volume, functional residual capacity, and total lung capacity, are measured with a spirometer. Most spirometers used today are computerized systems that utilize a differential pressure transducer to make measurements that are converted to volumes and flows. These volumes and flows are then displayed on a computer monitor in text and graphic form. The individual pictured here is undergoing a spirometry test. Wearing nose clips, she makes a tight seal with her mouth on the mouthpiece. The technologist then instructs her in performing a variety of maneuvers that will result in the measurement of the lung volumes and capacities. Typically, the patient will be instructed to breathe normally and then to take a deep breath all the way in. She will then be instructed to exhale completely. This can be done slowly for a slow vital capacity measurement or rapidly for a forced vital capacity measurement. Lung volumes and capacities are measured using a slow vital capacity maneuver, while airflows are measured using a forced vital capacity maneuver. Pictured here is a spirometry tracing showing all four lung volumes: tidal volume, inspiratory reserve volume, expiratory reserve volume, and residual volume, as well as all four lung capacities: inspiratory capacity, vital capacity, functional residual capacity, and total lung capacity. We will discuss each of these separately. The tidal volume is the amount of gas an individual inspires or expires during normal, quiet breathing. So as you sit there breathing normally, in and out, that's your tidal volume. Normally that volume is 3 to 4 mLs per pound or 7 to 9 mLs per kilogram of ideal body weight. which is about 8 to 10% of the total lung capacity. The inspiratory reserve volume is the amount of gas that an individual can inhale above a tidal inspiration. So as you're sitting there and you make a normal tidal inspiration and then take a deep breath all the way in as far as you can, that's an inspiratory reserve volume. Normally, it is 60% of the total lung capacity. The expiratory reserve volume is the amount of gas that an individual can exhale beyond a tidal expiration. So as you're sitting there and exhale normally, and then push all the gas out that you possibly can, that's your expiratory reserve volume. Normally, it is 20% of the total lung capacity. The residual volume is the amount of gas remaining in the lungs after a maximal expiration. So as you're sitting there and you blow all the way out as far as you can, the amount of gas that remains in the lungs is the residual volume. Normally, it's 20% of the total lung capacity. The residual volume cannot be exhaled, so it cannot be measured directly with spirometry. The inspiratory capacity is the amount of gas that an individual can inhale starting at a tidal expiration. So as you breathe out normally and then take a big deep breath all the way in, that's the inspiratory capacity. Therefore, it includes the tidal volume and the inspiratory reserve volume. You might take an inspiratory capacity before jumping into the water so that you can stay underwater longer. Normally, it is 60% of the total lung capacity. The vital capacity is the amount of gas that can be exhaled after a maximal inspiration. So what you would do is take a big deep breath all the way in and then blow it all the way out as far as you can, and that would be a vital capacity. Therefore, it includes the inspiratory reserve volume, tidal volume, and expiratory reserve volume. The functional residual capacity is the amount of gas remaining in the lungs after a tidal expiration. It includes the expiratory reserve volume and the residual volume. Normally, it is 40% of the total lung capacity. The FRC cannot be measured with spirometry because the RV cannot be exhaled and therefore it cannot be measured directly. The FRC has to be measured indirectly using helium dilution, nitrogen washout, or body plethysmography. The total lung capacity is the amount of gas in the lungs after a maximal inspiration. So if you take a big deep breath in and hold it, the volume of gas in your lungs is the total lung capacity. Therefore, it includes the inspiratory reserve volume, tidal volume, expiratory reserve volume, and the residual volume. Normally, it is about 6,000 mLs for adult males and 4,200 mLs for adult females.
All right, let's turn on that. Go ahead and breathe. So right now, our subject is breathing to the room, breathing normally. Now the subject is breathing into the spirometer, the classic water-filled spirometer, breathing normal tidal volumes. We'll take one more tidal volume. And I want you to take a big breath in, pause, and blow it out as hard as you can. Blow, push, 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 push. Okay, come off. Okay, take a break. We got time. I'm going to give you an obstruction now.
Okay.
So there's the mouth piece that she breathes through. This is the obstruction. So air will be reduced to not this area, but this hole right here is what she'll be breathing through. So there's the obstruction. Most people do hate the obstruction. Go ahead and breathe. Breathing to the air. Now again, small volumes. Now breathing to the device with the obstruction in place. Normal tidal volumes with the obstruction. Now one more. Now inhale all the way. Give it everything you got. Pause, blow it out, push as hard as you can, push it out, push, 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 and come off. What's noticeably different with the obstructed breathing is the FEV1, the forced expiratory volume in 1 second. You can see again if we're coming over approximately 32 millimeters, which would be that line in fact, in 1 second, that's how much of the total vital capacity was pushed out in 1 second and obstructed versus how much was pushed out in the unobstructed. This is the main value that changes with the obstructive lung disease.
The branching air passageways of the mammalian lung end in clusters of tiny sacs called alveoli. The alveoli are the sites of gas exchange. Each alveolus is surrounded by capillaries. Oxygen and carbon dioxide can diffuse rapidly across the thin walls of an alveolus and its adjacent capillary. Blood entering lung capillaries from the systemic circulation is low in oxygen and high in carbon dioxide. Air inside the alveolus is high in oxygen and low in carbon dioxide. Each gas follows its concentration gradient. Oxygen entering the bloodstream binds to hemoglobin in red blood cells. Carbon dioxide entering the lungs is exhaled.
Lungs are infoldings of the body surface that are used for gas exchange. The lungs of mammals are divided into tiny sacs called alveoli. Deoxygenated blood flows through the capillaries that cover each alveolus. When air enters the respiratory tract and reaches an alveolus, oxygen diffuses across the respiratory surfaces. The blood is now oxygenated as it leaves the surrounding capillaries. The alveolus functions as an interface between air and blood. That interface consists of a layer of epithelial cells, some extracellular fluid, and the wall of a capillary. The capillary wall is so thin that oxygen and carbon dioxide readily cross it. The bloodstream distributes the oxygen from the lungs to the tissues that need it. The bloodstream also delivers the carbon dioxide from the tissues that generated this waste product to the lungs for expulsion.
Fresh air entering the lung carries oxygen with a PO2 of 160. The presence of moisture in the lung results in reduction of the PO2 to 104. Fresh air entering the lung carries carbon dioxide with a pCO2 of 0.3. The carbon dioxide delivered to the lung from the blood raises the pCO2 to 40. At the arterial ends of the pulmonary capillaries, oxygen diffuses from the air in the alveoli into the blood, and carbon dioxide diffuses from the blood into the alveoli because of differences in partial pressures. As a result of diffusion at the venous ends of the pulmonary capillaries, the PO2 in the blood is equal to the PO2 in the alveoli, and the pCO2 in the blood is equal to the pCO2 in the alveoli. With no differences in partial pressure, there is no more net movement of oxygen or carbon dioxide. Oxygen diffuses out of the arterial ends of tissue capillaries into the tissue fluid, then into the cells, and carbon dioxide diffuses out of the cells into the tissue fluid, then into the blood because of differences in partial pressures. At the venous ends of tissue capillaries, the PO2 in the blood is equal to the PO2 in the tissue fluid, and the pCO2 in the blood is equal to the pCO2 in the tissue fluid, resulting in no more net movement of oxygen or carbon dioxide. The blood now carries the oxygen and carbon dioxide to the lungs. In the body, all of these exchanges occur simultaneously.